Hello and welcome to the immune system video for our AP Bio exam review video series. This is the longest video in this unit just because there's so much detail with the immune system. All right, you need to know several elements of the innate immune response. So the first thing we're going to do is just talk about what the difference is between the innate immune response and then adaptive immunity. And so in the innate immune response, this is present in all animals, whereas adaptive is only in vertebrates. And the innate immune response, that is a broad range of responses that are not specific. This is my example where I was just going out in my yard and spraying weed killer on everything, and it was killing all of my plants, even the stuff I liked. The thing about innate immunity is that this is the stuff we don't really think about because this is the stuff that prevents us from getting sick in the first place. You don't notice your innate immunity keeping you healthy. All you notice is when your innate immunity fails and you have to get into adaptive immunity. Because adaptive immunity is a slower response. It's very, very specific to saying, okay, what are you sick with and how are we going to attack that? Versus innate immunity is just very widespread. Hey, there's something here and I'm just going to kill everything. And so you don't really notice when innate immunity is doing its job. You just notice that you're not sick. And so that's what this is saying. It's a rapid response. So innate immunity really has internal and external defenses. So your barrier defenses are the things that just keep pathogens out. Your skin, your mucous membranes, the things that you secrete, your digestive system, all of those kinds of things that just keep stuff out of your body that shouldn't be there. If stuff gets in, your innate immunity does have some responses as well. And again, non-specific defenses. And that's these right here, your phagocytic cells, your natural killer cells, antimicrobial proteins, and then your inflammatory response. And so these are the things that are very general, but are inside. The specific stuff is the adaptive immunity, which we'll get to a little bit later. So phagocytosis, this is an aspect of your innate immunity where we have our phagocytic cells, and these are all white blood cells, your phagocytic cells that find pathogens, envelop them in a vesicle or a vacuole, and then have a lysosome that comes together with it. They fuse, the lysosome then releases all of its enzymes into this other vesicle, and everything is destroyed, and then debris is released from the cell. And so this is the whole idea behind finding a pathogen and destroying it. And that is very general phagocytic cell, part of your innate immune system. How do these phagocytic cells actually find the stuff that's floating around? They use these things called TLRs. We talked about TLRs in class just to say that different TLRs are looking for different things. And so like, here's an example. This is dsRNA. That's double-stranded RNA. There is no good reason for you to have double-stranded RNA in your body. And so this particular TLR is a membrane protein that's just sitting out there looking, and it's just looking for double-stranded RNA. And if it finds it, then wherever it finds it, it says, oh, we need to get on the alert and engage our innate immune response. And that can be eating the pathogen, consuming the pathogen by phagocytosis. That can be initiating an inflammatory response or any of our other innate immune system responses. And so TLRs are they're called toll-like receptors. The TLRs are just looking for macromolecules that should not be in your body because they are only present on things like bacteria, and then responding to them in an appropriate way. Okay. Inflammatory response is also a part of your innate immune system, and what that's just doing is releasing chemicals after an injury. In this case, we're looking at a splinter, releasing chemicals that would cause antimicrobial peptides to come into the tissue in order to prevent infection. And so anytime you have any kind of severing, here's your skin and there's your barrier. Anytime you have a breach in that barrier, that's when we are now going to introduce bacteria. Bacteria are everywhere, you can't get away from that. So you have mast cells. Mast cells are releasing histamines, that's what these red things are, as well as cytokines, and those are recruiters for other neutrophils, here they are, Okay. What do these signaling do? They recruit other blood cells, white blood cells, but then as well they dilate this blood vessel. See how it's thicker here. And then they increase porosity. See how large the hole is here versus here, encouraging these cells to come to the site of the injury. Once those cells are at the site of the injury, what we end up with are these phagocytic cells that come in and literally engulf and destroy the pathogens that I showed you a couple of slides ago. So this is your inflammatory response. Inflamed skin tends to be red and warm as a result of this increased blood flow. All right, so now we're going to move into your acquired or adaptive immune response, and so that's going to involve the B cells and the T cells. All of them are produced from stem cells, adult stem cells in your bone marrow. The B cells stay there for activation, whereas the T cells go to the thymus. The B cells will go on to rid body fluids of pathogens. The T cells will go on to rid body cells of pathogens. We have to do both because almost always pathogens that are in your body are using your cells to reproduce. And so if you rid your cells of infection, there's going to be some that was already moving cell to cell, and so the body fluids would then be infected even though the cells were clean. Or if you only cleaned up the body fluids, then the cells themselves would be producing more. So your immune system has to clean up both your body fluids and your body cells when you're sick. And so we use two different kinds of immune system cells in order to achieve that. You must know how antigens are recognized by immune system cells. So here we are, we're looking at a B cell antigen receptor. And so here's a B cell. We're going to remember that every B cell and T cell has millions of these receptors on the outside, but they're all exactly the same. And so each receptor looks for the exact same protein or marker for a pathogen. And so we have our constant regions and we have our variable regions. And at the very end of this variable region is where we have the antigen binding site. These things are locked together by disulfide bridges. So this is something that has quaternary structure. I guess that the disulfide bridge is actually a tertiary structure. But we're ending up with a couple of different strands of proteins that all come together to form this one protein. The T cell antigen receptor looks a little bit different in just that instead of being Y-shaped, it's just linear. But the variable end is still here, and we still have the constant N, and it's still locked together by that disulfide bridge. So then how do they actually recognize things? Well, pathogens are very, very large. And remember, these are just proteins. So these are just individual molecules. And so instead of looking for the entire pathogen, each of these membrane proteins or each of these antigen receptors is looking for a very small part of that pathogen. And that is called an epitope. And so the epitope is just this little chunk on the end of the antigen that's connected to the pathogen. And so the B cells actually bind, it's a conformational change or conformational recognition that are looking for the proper shape to connect to that particular antigen or pathogen rather. And then once that has bound to it like this, then it can be marked for destruction or being disabled because of conformational change. But it's all based on conformation, right? The B cells and the antibodies are all matched up by the same shape, conformational shape, and that's what allows them to identify pathogens. You need to know the differences in humoral and cell-mediated immunity. We're going to remember that humoral immunity, that's your body fluids. So first we're going to look at T cells. So T cells are the ones that are going to kill infected cells. And so this is just showing you that the effector cells here are the cytotoxic T cells. What they're doing is they're finding a cell that is infected. And the way they're finding a cell that is infected is that the cell itself has to declare itself as sick, and it has to literally flag a T cell to come over and destroy it. And so it takes a chunk of the antigen that has infected the cell, presents that thing on a class 1 MHC molecule, and then the cytotoxic T cell recognizes that antigen and then literally perforates the cell membrane and kills the cell off. And so in this case, the cytotoxic T cells aren't just going around killing all your cells. They're only going around killing the cells that have this antigen fragment presented to it on that MHC molecule. B cells are not altogether too different, except that B cells aren't actually looking for cells. So B cells are supposed to be cleaning up the body fluids. And so what happens here is we have, again, that helper T cell, and we'll get to that helper T cell in just a second. The helper T cell activates the B cell, but what the B cell does is, instead of looking for pathogens or whatever, all it does is produces lots and lots and lots of antibodies. And those antibodies are specific to the pathogen. Those antibodies then are floating around the body fluid, binding to anything that matches this conformational change or conformational shape at the end of the protein, and marks it for destruction. And so the plasma cells in this case are producing these antibodies. It was said 20,000 a second for 4 or 5 days. So massive quantities of these antibodies that are just released into the blood. And then these guys are the things that go around and actually look for pathogens that are floating around. Now, you need to know why helper T cells are central to the immune response. So earlier I talked about what do the B cells do in order to get humoral immunity, and what do the T cells do in order to get cell-mediated immunity, and we kind of skipped over this. How did they get activated in the first place? Well, here's what happens. This is an antigen-presenting cell, and we talked in class about this being a dendritic cell, and the dendritic cells were the link between the innate and the adaptive immune system. But the dendritic cells, they take in these pathogens, they destroy it, but then rather than just getting rid of the excess, they actually present a chunk of that pathogen on a class 2 MHC molecule, and that is what we're going to use to find the right helper T cell. And the right helper T cell is the one that has the proper conformation of this protein, this antigen recognizing protein. And so we go through, these guys hang out in your lymph nodes, this dendritic cell hangs out in your lymph nodes, and a whole stream of these white blood cells passes by over and over and over. And all it's doing is looking for the right shape to match up to this antigen. And so once that's found, then this goes through the process of clonal selection and finding the right B cell, finding the right T cell, and increasing those numbers hugely. And so helper T cells are what stimulate both sides of this immune system. You need to know how immunological memory is developed and what its impacts are. So this is an overview of the whole adaptive immune response. And we're going to remember that here's your antigen-presenting cell. And so those are your dendritic cells. It can also be your macrophages. But these cells right here are part of the innate immune system. But what happens is that antigen comes in, the innate immune system is doing its best to get rid of it. At the same time, it communicates with these helper T cells that says, hey, let's see if we can find the right kind of helper T cell in order to stimulate the rest of this. And so once that's happened, once the helper T cell has the right antigen with it, it then stimulates the B cells and the T cells. And then these guys are the effector cells, right? We have to have effector cells. The plasma cells, which secrete your antibodies, which defend against extracellular pathogens. And then we have cytotoxic T cells that defend against infected cells. And then, of course, we also have these memory cells. And here's why this matters. We have our memory B cells, our memory T cells, our memory helper T cells, and these guys live for a very, very, very long time, if not forever, in your body. And what happens is, should you be exposed to that same antigen again, rather than having to go through the process of finding the right helper T cell, this process can take as long as 3 to 7 days. We jump straight to this, where we go, oh, okay, straight back to these memory cells. We get a very quick response, and the reality is you have much more antibodies produced in a shorter period of time, and so you just don't get sick. This is the graph that shows you that. This is showing you, okay, you get exposed here to antibody that is blue, or to sickness implement that is blue. You get exposed to this antigen, and it takes 5 days before you're able to actually produce those antibodies. So that's how long it takes to find the right helper T cell, for the helper T cell to then stimulate the B cell to produce the antibodies. There's this delay. However, if you look at exposure, the second exposure to that same antigen, then it's literally same day that we're able to get response to those antibodies. Also, initially, the antibody concentration was this much initially. The second time it's this much, which means we're cleaning up your body fluids much, much, much faster. And so this is specific to something you've been sick with before. And so here, you got sick with it once, you won't get sick with it again, even if you're exposed to it, but that only works for that particular antigen. You get exposed to a second antigen, and once again, you have to go through that process of finding the right T cell to stimulate the right B cell, to stimulate the right T cell, and et cetera, et cetera, et cetera. And so you have to go through this primary immune response again with that new antigen. So just understand that the secondary immune response is more dramatic and faster than it was the first time. You must know how immune self-tolerance develops and the problems that may arise when it does not. So this is a diagram right out of your textbook. I didn't show it to you in class, but it summarizes really, really nicely, at least as far as the B cells are concerned. And of course, the same thing's happening with the T cells. But adult stem cells in your bone marrow are what are causing all of these other white blood cells to develop. And this is coming from gene rearrangement. Remember, we used the enzyme recombinase to recombine the DNA from these particular genes. And then we have to go through this process of self-tolerance. And what that means is before these cells are going to be ready to leave the bone marrow, they are going to be tested against your own cells. And if they fail the test, as in they respond to your cells, then they go through apoptosis. The ones who are able to recognize your own cells and not respond to them are then released into the blood. And then when they're stimulated by an antigen to go through clonal selection, that's how we end up with all of the cells in the process that I talked about earlier. But just understand that we start with the adult stem cells, we have recombination of the genes, and that's part of the maturation process. Once they're mature, then we test them for self-reactivity, and if they fail that test, we kill them. If they perform appropriately, that's when they are actually released to the blood. It seems like that should be a perfect system, but it's really not. We have the incidence of rheumatoid arthritis is fairly significant. And what happens in many cases, I gave you lots of examples in class, is that the immune system passed the test of self-tolerance in the bone marrow or in the thymus, depending on which kind of cell it was, except it was a false reading. It turns out that those immune cells are still responding to some of the molecules that are in your body. And so that's what rheumatoid arthritis is that the immune system is attacking the cartilage in the joints, and it causes, this is a very severe case, but it causes all kinds of problems. And so it's problems of self-tolerance that cause all kinds of issues, and they're called autoimmune issues. And so type 1 diabetes is another example of that. They're actually fairly common. So the system, while good to keep you healthy, is certainly not perfect. So anyway, that's the end of our immune system video. Thanks for watching. I'll see you in class. Have a good day.